Hello everyone. Today let's discuss about platelets, which are also called as thrombocytes. These are small colorless cell fragments in our blood that form clots and stop or prevent bleeding. The average platelet count in humans ranges from 1,50,000 to 4 lakhs per cubic mm. The lifespan of platelet is approximately 10 days, and the average adult must produce approximately 100 billion platelets each day to maintain a normal platelet count. Now, let's talk about thrombopoiesis, that is, production of platelets. Platelets are formed from megakaryoblast, which is a precursor cell to a promegakaryocyte, which in turn becomes a megakaryocyte. During thrombopoiesis, the megakaryoblast is formed from the colony forming unit megakaryocyte, that is CFU cell, which in turn is formed from the colony forming unit spleen, that is CFUS. The primary regulator of platelet production is thrombopoietin, produced primarily in liver, kidney and bone marrow. Now let's talk about hemostasis. The term hemostasis means prevention of blood loss. Whenever a vessel is severed or ruptured, hemostasis is achieved by several mechanisms like vascular constriction, formation of a platelet plug, formation of a blood clot as a result of blood coagulation, and eventual growth of fibrous tissue into the blood clot to close the hole in the vessel permanently. Let's look on each of them one by one. Vascular constriction. Immediately after a blood vessel has been cut or ruptured, the trauma to the vessel wall causes smooth muscle in the wall contract. This instantaneously reduces the flow of blood from the ruptured vessel. The contraction results from local myogenic spasm, local autocoid factors from the traumatized tissue and blood platelets, and nervous reflexes. However, even more vasoconstriction probably results from local myogenic contraction of the blood vessels initiated by direct damage to the vascular wall. And for the smaller vessels, the platelets are responsible for much of the smaller vessels. The platelets are responsible for much of the vasoconstriction by resulting vasoconstrictor substance, that is thromboxane A2. The more severe a vessel is traumatized, the greater is the degree of vascular spasm. The spasm can last for many minutes or even hours. During this time, the formation of platelet plug occurs. Let's talk about that. If the cut in the blood vessel is very small, indeed many very small vascular holes develop throughout the body each day, the cut is often sealed by a platelet plug rather than by a blood clot. To understand this process, it is important that we first discuss the nature of platelets themselves. But now let's talk about blood coagulation in the rupture. The third mechanism for hemostasis is formation of the blood clot. The clot begins to develop in 15 to 20 seconds if the trauma to the vascular wall has been severe, and in 1 to 2 minutes if the trauma has been minor. Activator substances from the traumatized vascular wall, from platelets and from blood proteins adhering to the traumatized vascular wall initiate the clotting process. Now let's see the steps of clotting process. First is severed vessel. Then the platelets agglutinate. Fibrin appears, fibrin clot forms, and finally clot retraction occurs. Within 3 to 6 minutes after rupture of vessel, the entire opening or broken end of the vessel is filled with clot if the vessel opening is not too long. Now let's talk about the physical and chemical characteristics of platelets. Platelets are minute discs 1 to 4 micrometer in diameter. They are formed in the bone marrow from megakaryocytes. Platelets have many functional characteristics of whole cells, even though they do not have nucleus and cannot reproduce. In their cytoplasm, there are actin and myosin molecules which are contractile proteins similar to those found in muscle cells, and still another contractile protein, thrombostenin, that can cause platelet to contract. Residuals of both the endoplasmic reticulum and the Golgi apparatus that synthesize various enzymes and especially store large quantities of calcium ions. Mitochondria and enzyme systems that are capable of forming adenosine triphosphate, that is ATP, and adenosine diphosphate, ADP. Enzyme systems that synthesize prostaglandins, which are local hormones that cause many vascular and other local tissue reactions. So that was all for platelets, thrombopoiesis, and the physical and chemical characteristics. In the next video, we will talk about some pathology related to platelets. Till then, take care, stay safe. Like, share and subscribe to the channel. Thank you.